Okay, it is 1 o'clock. I believe we should start. And I've been asked to provide a little background about myself and how I came to be where I am today, and particularly how I got into astrobiology. And I guess back in high school, my inspiration to get involved with chemistry got started. As usual, there's the inspirational teacher, the high school chemistry teacher, who was also quite an outdoorsman. And that had an effect, although I didn't realize it at the time. Also, my older brother decided to get into chemical engineering, so the word chemistry kept popping up here and there. And my father worked for DuPont Corporation, which, of course, is very well known for their chemical research and their plastics and all kinds of other products. Better living through chemistry was their motto back then. It sort of fell out of favor, I think, in subsequent years. But anyway, it was a message that I had received at some level. So I majored in chemistry at Purdue University. And got interested in outdoor stuff as a weekend activity, which led to cave exploring, which led to an interest in geology. So I took a geology course and got very interested and was advised to stay in chemistry to get my undergraduate degree, stay with the core discipline, I was told. Very good advice. But then I went off to Indiana University to pursue an advanced degree in geochemistry. And that got me involved with lunar sample analysis, analyzing carbon, nitrogen, hydrogen, in the lunar finds and ultimately lunar rocks. And of course that obviously started the NASA connection. But while I was there, my major professor, John Hayes, was very much involved in biogeochemistry and very much in the issue of searching for life beyond the Earth. He was involved in much of the planning and preparation for the Viking mission. And so that naturally led to me eventually applying for a position at Ames Research Center to continue studying sort of carbon in the universe, which was another theme they were pushing in those days. And that got me to, basically exobiology, which was pretty much the forerunner for astrobiology. Interestingly, as you probably know, the word astrobiology was raised back in the 60s as an interesting name for the program, but some people thought that was too close to astrology to be comfortable. And it's interesting that concern wasn't raised so much in recent years, in the late 90s, when astrobiology finally did take hold as a name for the program. So anyway, down the years, I've been involved in carbon, nitrogen, hydrogen geochemistry of lunar samples, got involved with mid-ocean ridge basalt research with the budget of carbon coming up from the mantle along the mid-ocean ridge system, and actually performed the first accurate calculation of what that flux was, leading to the surprise that we're pumping out 100 to 200 times more carbon, CO2, into the atmosphere than all the world's volcanoes. So that was an insight. And then because of the interest in biology that first started at Indiana, I got involved in a microbial mat research program down in Guerrero Negro in Baja California Sur. And so I've been sort of around here, the bio and the geo parts. And then, of course, when the Mars program finally got a jump start in the 90s, I got involved in program planning for missions that would follow up on the Pathfinder mission, and that led ultimately to my involvement in the Mars Exploration Rovers, principally with the Spirit rover, and now more recently with Curiosity. Along the way, I've also was involved in the CRISM instrument team with Mars Reconnaissance Orbiter. So the latter part of my career, once these missions got going, really got me into the space exploration business, and that's sort of where I am today. Along the way, also, I was very much involved in the roadmap, Astrobiology roadmap, when it was first envisioned in 1997, and then I chaired the revision of it in 2003 and 2008. And of course, as you know, it's now being up again for revision. It's been 10 years already since we did our major revision in 2003. So I'm involved also in Mars program planning, trying to make sure that the astrobiology flavor is in there in reality and not just in name. And that's been actually a quite rewarding effort. As you know, we're really focusing on search for evidence of life now and habitable environments on Mars. So that's the overview, and I think the key point is to maintain a real core discipline, something like in chemistry, physics, or biology that really gives you those qualifications for research, but then also in astrobiology, of course, to get into the interdisciplinary aspects, because that's, as you know, where the big interesting questions are, not just with searching for life beyond the Earth, but in environmental questions and a lot of other things that face our society today. So we think that this is a great program, not just because space exploration is great, but because it really prepares people for the type of interdisciplinary perspective and capability that will help us address our big problems in the future in society. So I'm pretty upbeat about astrobiology, obviously, and that's a little bit of how I came to where I am today. So with that, I guess I'm open for questions. I see one here, and should identify the person, I guess. Kara Tucker asking this question. How do you find wavelength ranges for detecting biosignatures on extrasolar planets? Oh my, okay. Well, really, let me first issue the disclaimer. I'm not a spectroscopist, but what I can tell you is that these wavelength ranges that people focus on are pretty much defined by the kinds of hardware that need to be optimized for that particular range. And so, visible near infrared tends to be in one domain, and of course, is associated with that, you have the the types of molecular transitions and vibration states, I guess, really that would create features in that range. And so that's one bin. And then when you get into the mid to infrared, you're really talking about a different type of sensors, and of course, you're now looking at more fundamental molecular vibrations, which tell you, for example, a lot more about cations, or anions, I should say, what the anions are associated with the molecules that you're seeing out there. And then, of course, there's people who even, are interested in doing microwave surveys, the whole study, search for evidence of intelligent life. So it's pretty much the wavelength ranges are dictated by the particular instrumentation and therefore the observing telescope that one would need to deploy. But again, each of these ranges is very complementary to the other, and we always would argue that if you can have two complementary wavelength ranges sort of both focused at looking at atmospheric composition or surface composition or biosignatures, that really helps to constrain interpretations the best. So to first order then, the answer is it's oriented based on the type of instrumentation required, and then secondly on the types of molecular features that you're associated with those, and then the science flows from that. Okay, Shauna Kendall, what instrument package would you like to see on a rover searching for life on Mars? That's a very interesting question. With the basic approach to searching for evidence of life on Mars is first to locate the haystack before you search for the needle. In this case, the haystack is the habitable environment. And given that the surface of Mars is equal to the area of all the continents on the Earth, that's a lot of real estate, and a lot of that real estate we think is not particularly promising to search for life, and that's simply because it's volcanic lava flows, features that are pretty much dominated by surface processes on Mars that have pretty much dominated over the last 2-3 billion years. And as you know, a very, very dry place, not so much a problem with the cold as it is with the very profound dryness. So one area of interest today might be at the margins of the polar caps, but pretty much, we're talking about looking for, our focus in the program really now is looking for past life, and that would be life probably in the deep past. And so for this, we need instrumentation on a rover that tells us about the presence of water, the extent to which water might have interacted with the rocks, and then of course with that interaction you get the nutrients for life, and then you have the question of energy sources. So you pretty much have to do a geochemical type of analysis to see if you have preserved evidence for what might have been a habitable environment. And even if there is life on Mars today, if there's some feature at the surface that could have preserved evidence of it, you still have to do this kind of a mineralogical compositional study to see if you're really in an area that has the capacity to preserve something that would have represented life. So basically a geological prospecting package of capability of camera, obviously. You've got to have mobility in camera and then the ability to actually do an elemental abundance measurement, the kinds of thing, and of course mineralogy. And that mineralogy could be done a number of different ways. Spectroscopy, of course, we'd love to do mineralogy with the X-ray diffraction capabilities of the Chemin instrument that's flying on Curiosity right now. So I would love to have that capability to determine what kinds of minerals, particularly clay minerals, might be there, because some are better at preserving evidence of life than others. You'd like to have some kind of a capability to look at the volatile and organic fraction. One of the best instruments for that is a mass spectrometer. I'd love to have that pretty much embodied with what we're seeing with SAM in the Curiosity mission. We're really coming to believe that the package that is on Curiosity is really ideal in many respects. What I'd like to add there in terms of sample preparation, though, would be to be able to grind into a surface of a rock so that we could do a better interrogation of fresh material. We don't quite really have that capability with Curiosity, but I'd love to see that in there. And what we're really looking for are not the definitive evidence of life, but what we would call potential biosignatures. And that any organic matter is a potential biosignature in my book, even if it's meteoritic, because that means you found a place that not only was habitable, but was able to preserve evidence of organic matter if it had been contributed there. And so we're looking for these potential biosignatures, and this could include morphologic features. It could include certain types of minerals, such as magnetite and so forth. Sometimes are known to be produced by microbes. But really for us, the ability to detect the presence of organic matter would really be high on my list for capabilities of a rover. And so really much of what I would put on that list is what Curiosity has, plus that rock cleaning tool. Okay, I'll leave that for the moment. We can always come back if you want more of a detail in the answer. Okay, Julia Des Marais, describe some of your most memorable moments with your involvement with the Mars program. Well, anytime you're talking about Mars missions, and especially landers, the memorable moments are the landings. As you probably all know, the single most dangerous part of the mission that a rover has to experience is that entry, descent, and landing at Mars. The U.S. has now developed quite an amazing track record in this regard, and so we really seem to have sort of caught the magic potion here that really addresses that very dangerous part of the mission. But you know, we have to just realize that every time we send a new rover. So just with everybody else, I mean, it was a global experience this last time when Curiosity landed so safely, and it's landed so perfectly in many respects. So for me, that's a highlight. The other highlight is, like for example, on the Spirit mission, Mars Exploration Rovers, when we landed and we saw nothing but evidence of lava flows all around us and meteorite impacts and just dry, dusty, windy deposits, and just wondering, oh my gosh, is this the mission? You know, obviously we'll analyze these lava rocks, you know, the basalts, but you know, we really landed here because we thought Gusev Crater was a lake bed, you know. And so for us, I think it was just thrilling to take this roll of the dice to drive over 2 kilometers, 2.5 kilometers away from our landing spot. And you might remember that we only promised the world these rovers would drive 600 meters. So we said, well, let's roll the dice and let's go to these hills that we see over there. And so for me, it was incredibly exciting that they turned out to have been very altered by water. For all we know, they really do represent some aspect of the lake bed that was there. And so this was just tremendously rewarding. And then another follow-up with that mission was very exciting, was when we went down to this place called Home Plate and found first sort of acid sulfate deposits, and then also the pure silica that really told us that not only was there volcanism here in water, but hydrothermal activity, that this, for all intents and purposes, could have been a habitable environment. And by golly, we just went and found that all on our own. We went to the right place. We made the right measurements. Even with a mission that, you know, has understandably a more restricted set of capabilities than Curiosity does, still we were able with that mission to make a very satisfying determination that we were in a hot spring environment. And so for me, you know, it's just anything that's the end of a long effort and uncertainty and danger and all of that turns out well, it's got to be right at the top of rewarding. And of course, after Spirit sort of passed into the history books, for me, the other rewarding thing was that opportunity was able to get all the way down to Endeavour Crater. I mean, Spirit sort of warned us, you know, these wheel actuators, they're not going to last forever, and yet here's opportunity chugging along. And for those of you who don't know, it'll be very exciting this summer when it looks like it's going to pass the Lunicod driving record and have the record for the longest traverse on another planet by human technology. And so that'll be exciting as well. Just the fact these rovers have lasted so long, I think, is a major excitement. Okay, let's see here. How did our understanding of surface mineralogy petrology improve or change after the findings of MRO? Well, boy, a lot of things came out of the MRO. The Mars Express mission, the Omega instrument, really made the initial and very important, exciting discoveries of phyllosilicates or clay minerals on Mars, clearly demonstrating that yes, water wasn't just running across the surface episodically, but it was hanging around long enough to really alter the rocks in ways that we're familiar with on the Earth. What was amazing about MRO, of course, there's a lot of instruments, and I'm sure I'm doing some of them injustice by focusing on just the CRISM instrument, which did the mineralogy, and of course the HiRISE camera, which really gave us those beautiful high-resolution images, including color of the surface. So, I mean, in the end, you have to relate composition to features that you can see on the ground, that you would see if you were just walking around. And those two instruments in combination have really done that. CRISM, or the Mars Express Omega instrument, discovered the phyllosilicates, but CRISM mapped them at a resolution that was really able to allow us to reconcile what we saw there with what we see with the HiRISE instrument, and to really get to the point here, to do this at a scale that is the scale of exploration of a rover on the surface. And so with those two instruments, orbital instruments working in concert now with these rovers, we are really, doing a rover mission. We are doing surface exploration from all these different perspectives that are just working wonderfully together. And so just for example now, we're on the verge of driving Curiosity to the mountain, Mount Sharp. And my gosh, we have a view of the road ahead like we never could have dreamed. I mean, we've got, better than, half-meter resolution or 30-centimeter resolution on the surface with HiRISE. We have compositional information of that hill from CRISM. And it just harkened back to the story I just mentioned about Spirit when we looked those hills over there and thought, oh, geez, what's to lose? Let's go over there. I mean, if we know what we know now about those hills, if we'd known that at the time, we would have gone straight for certain places and really been able to discover a lot more. So MRO has just added this dimension. And of course, in terms of site selection, where do we go next? I told you about how huge the surface area of Mars is. The idea of selecting future sites now is just at a whole new plane of existence because of the imagery of HiRISE coupled with the mineralogy capabilities of the assets that we have up there. It's just been transformational, and we now have this idea about a pervasive presence of water. Early on Mars, these phyllosilicates now with this improved resolution are global in distribution, especially at low latitudes, and it's a whole new view of Mars. And of course, it's just made it much more exciting. Now our problem is, which of these great places do we go to in the future? And at some point, scientists are going to be divided in their opinions for good reasons, and the engineers will just pick the safest place. So, in a way, for scientists to have a control on the future landing site selection process, they're going to have to think through about how they're going to prioritize amongst a bunch of very promising places, as revealed by HiRISE and MRO. Okay, let's see. Let me switch to another person here, Svet Skoldier. Piggybacking on the previous instrument site question, what's your vision for a Mars sample return in terms of lifetime and instrument suite, and is this the way you think NASA is taking that plan? Well, I have to be careful here because I happen to be on the science definition team for Mars 2020, okay? But I think it is safe for me to say that now that we've done a few rovers, the two MER rovers and the Curiosity rover, that we've identified a certain suite of measurement categories that are very robust. I mean, we clearly want to have a camera, okay? We clearly want to have some way of doing a chemistry, as we call it, or elemental abundance measurements. We need to interact with the surface of a sample, a rock, or whatever, in some ways to give us a better exposure, since everything seems to be covered with dust. And we need to approach mineralogy. And I guess the other thing I'll say that's not going too far is that you need to be able to address these things both as you're looking out in the field there, tens to hundreds of meters out, not only with respect to what you see with your eyes, but what you might be able to infer about mineralogy. And then, of course, you want to be able to also have those kinds of capabilities when you get up and close and personal with a sample. And so just the basic, visual observation, compositional information that you would need at the sample scale to make selections for samples, we just need that type of capability. So maybe a key answer to your question is that a Mars sample return mission has to be a mission much like the Mars rovers were in its own right. It's got to be a mission that's exciting as it's going along, picking out the samples, but also just making those contextual observations. And then it's it should be collecting these samples. And the key thing is that we're going to put these samples in a little container. We're going to abide by all kinds of rules in doing that, and not just planetary protection and all that, but also for the science integrity of the samples. And we have to be prepared for the possibility that it might be a few years before these samples get picked up. And the principal reason for that's political, frankly. I mean, it's when are we going to have the budgets, and how are we going to be fair to the other parts of the solar system with respect to when we maybe go to Europa or Titan or some outer solar system destination. So there's a lot of uncertainty about the timing of the return of those samples. So we have to be very careful about how we package and then store those so that they have good integrity when we finally get around to bringing them back. So these are all examples of things that we have to worry about, the sizes of the samples. The Mars Exploration Program Analysis Group, MEPAG, I was chair of that for about 2.5 years. If you go on that website, just type in MEPAG and it'll pop up as one of your selections in Google. There's a lot of documents in there that address this whole issue of what's the best way to do the samples and so forth. This is public information. This was the source of information that our science definition team very much took to heart in our deliberations. And that's probably a very detailed answer to your question. Let me circle back up to Kara here. What kind of clay minerals are best at preserving signs of past life on Mars? Okay, good question. Basically, the reason that you wash your clothes is that you're putting a detergent in there to separate clay minerals from organics, which happen to be your clothing. And so there's this intimate love affair between clay minerals and organics. And it's been found that principally the very high surface area of clay minerals are very fine grained, typically a lot of surface area for their volume. All that surface area there's an affinity with organic matter to that, so that the finer grained minerals, ones of course that are forming in water where the organic matter is present, are the types of minerals that are going to be the ones that we want to look for. So first off, we want to find clay minerals that as much as we can determine were formed in water, or it had a lot of exposure in water prior to their deposition, and that then these were preserved in a way that that the organic matter, if it was associated with them, would be preserved. And so, of course, as many of you know, the biggest problem we face there is oxidation on Mars. You look at Mars; it's oxidized at the surface, that nice red-orange color, and so we were very excited in the Curiosity mission when we got over there to Yellowknife Bay and we drilled into a surface that was red at the top, and suddenly we get into blue-gray kind of material coming up as the drill is going into the rock, and of course the material that went into the spacecraft for the analysis by Kemen and Sam also had that wonderful gray color, which said, hey, this stuff, my gosh, we were able just to go a few centimeters into the subsurface of this pavement and get material that has not been oxidized. And so it's not just the clay mineral, but the clay mineral whose iron or whose associated minerals are giving you an indication that this has not been oxidized. And so by implication, that deposit has been fairly impermeable. I mean, as I just said, we're only 2 centimeters from the surface, and yet you don't get the oxidation that you're seeing on the surface. And so the type of clay mineral, the type of clay deposit that is impermeable, obviously plays very well into the preservation aspect of our concerns. So you want an environment that was habitable, and that you could make the case there could have been life there, but then also an environment that allowed a sediment to be deposited and preserved that retained that record. And so those are the types of clay minerals that we're looking for. And I keep mentioning clay. It's 70% of where the organic matter is preserved in the crust of the Earth. So that's why we're so excited about finding these clay deposits. But sulfate deposits and carbonate deposits, and of course silica deposits, are also very good at preserving organic matter. But volumetrically, they're not as significant on the Earth as on Mars, but we do have sulfate deposits on Mars. I think our biggest concern is to be able to demonstrate somehow that that deposit was not oxidized. There wasn't an oxidation problem that happened after it was deposited, if indeed it had organics in it. So this is kind of the thinking we always go through. You know, the minerals that tell you it was habitable, but also the minerals or the state of the samples that tell you that they've been good repositories for preserving things. And then you're set up, I think, to have the most effective search for biosignatures. Okay. Shauna Kendall, in your opinion, what are the big questions that need addressed in astrobiology, terrestrial and or beyond? Well, let me start with the biggest question, and that is, of course, the origins of life. You know, I've often said, and I still believe, that probably the single most challenging question in science is to understand how life can begin. And I should partition that into two aspects here. One is, how could life begin in the universe? And then the particular case of how did life begin on the Earth? Okay. And curiously, the question of how could life begin anywhere in the universe is an easier question, because that is a question it's more deterministic in the sense of the kinds of chemistry and physics involved and the types of environments that planets or whatever could provide. You know, in principle, if those things are all in alignment in a certain environment, life could begin. And that's sort of a deterministic view, I realize, but it's something you could go and conceivably simulate somehow. You know, either in what do you call silicon in silico, or in vivo, in vitro, in the lab, or whatever. The harder question is, how did life begin on the Earth? Because that depends on what the particular circumstances were. It depends on environmental things that haven't been preserved. Okay, and so the point is that the Earth is so geologically active and life has been so dynamic in it and evolved that the traces of its particular origins might be forever not obtainable to us. So just understanding in general how life could begin is an easier question. Anyway, I think that's really the top question. And for me, it's easy to understand how life can persist if you know that you've got a source of energy and you're using that to maintain the kind of molecular and functional order that's needed to replicate and do all the things that keep cells going. You can calculate thermodynamically, yeah, that makes sense, it could happen. But then the question is, how does it start? How do you take the energy source and couple it to basically what amounts to an increase in information content of a molecule, of a structure, and of course, as you know, that's going against the second law of thermodynamics. So how do you push entropy back a bit to take energy in and begin to build up the kinds of information that meets the threshold of what would be something that could self-replicate or persist? And so I think, given all the wonderful research that's been done on RNA and molecules and some of the simplest molecules it could be, that's wonderful stuff. But they need to address how do you input the energy in the environment to that process to get it propagating. To me, that's a tough problem, and it's right at the top. The other key thing really relates to Mars exploration, and another big question, and that is, what really were these early environments? And the beauty of Mars, as you probably know, is that it's got this early record that's been preserved. I already just alluded to how that earliest record of environment on the Earth has been destroyed, but on Mars it's been preserved. What's just the most effective way to get at that? And what are the key aspects that we should be measuring that really relate to this question of the environment suitable for the origins of life? And I make another important point here about habitable environments. There's two flavors of these potentially, in principle. There's the habitable environment where if you sent something there today that was alive, it could survive. Then there's the planetary environment that could support the origins of life. And it's not clear to me that these are synonymous. Okay, for example, it's not clear to me, we could argue this, that the environments that you see on the Earth today are the best for the origins of life. Maybe not, maybe so. So there's two flavors of habitable environments that we have to keep in mind. So how do we sort this out, and particularly how do we go to another planet like Mars or Europa or Titan and really establish the information we needed to know about what that early environment was and how it relates to this origins question? So those are some really important things. And then, of course, the same thing applies to the early Earth. We know we have evidence of life certainly back to 3.7 billion years ago. Some people would argue older. But these rocks have really been tortured, and there's just so much more we'd like to learn about the nature of life at that time. But that requires the kind of detailed information that you find in organic molecules. And if we've really done all we can do in looking for those early evidences of life in the Archean record, rocks that are older than about 2.8 seem to be particularly well fried, and but yet it's not clear that everything has been just totally destroyed beyond recognition. So how can we get at that more effectively? By deep drilling projects, places just really go for where we think the best rocks are going to be, using the latest technology at the microscale, inclusions in rocks and all of that. And then, of course, the other major approach, which is the molecular approach of reconstructing early life capabilities from resurrection of genomes and enzyme capabilities. I think another area where astrobiologists could play a great role is what's the family tree of key enzymes in life, not just the DNA, the big tree of life based on 16 S-ribosomal RNA, but also the tree of various protein life. And as we know, that can sometimes punch further back even than some of the RNA DNA records potentially. And how does that help us understand just gene transfers and just the whole fabric with which evolution unfolded over billions of years? And so certainly astrobiologists having that desire to understand origins, I think, are going to bring an important flavor to the research labs of molecular biologists to really focus on our origins as preserved in the molecular record. And then, of course, extrasolar planets. I think, just like astrobiology, I think has played a key role in getting the Mars program restarted in the 90s, and also, a lot of what I've talked about earlier. I think we really need to take up the flag, to take a leading role to try to get the terrestrial planet finder type mission on the books. Again, that's a budgetary issue. It's not an inexpensive mission. But fortunately, Kepler lived long enough to show that there's just a lot of planets out there. We can really think about designing telescopes to analyze atmospheres, focusing more on the capability to analyze the nature of the atmosphere rather than just also trying to find planets. The planets now seem to be so widely distributed, and particularly around G-type stars like our own, that I think we really now can develop a major argument for moving forward on a terrestrial planet finder type mission. So what are the steps needed to get to that? And that's a challenge for the astrobiology community to pitch in on. Okay, well, those are some big questions I think need to be addressed. Let me see, let's move on to the next one. What do you feel has been your biggest contribution to the field of astrobiology, and what do you think has been the most surprising or impacting discovery in the field of astrobiology in the last 10 years? Well, I guess my biggest contribution to the field of astrobiology might have been earlier work I did on the carbon cycle. I mentioned calculating the flux of carbon from the mid-ocean ridges and trying to understand carbon on the Earth, not just in the context of organic matter that life makes and preserves in rocks and the atmosphere and the oceans, but also the whole cycle of carbon that reaches down deep into the Earth. And of course, as you know, a very important part of the story is how things have changed over billions of years. And there was much hotter in Earth interior in the early days. The mantle of the Earth was probably much more engaged with the surface environment back then than it is today, although it's obviously still very important today. But the point is, this really shaped the nature of that early environment. And so I think the work I did years ago on looking at carbon in early rocks, trying to calculate fluxes and so forth, provided a little bit more perspective and background for just really understanding what type of an environment the early Earth had. I think another contribution I've made was to sort of help to coordinate over the last some 25 years the microbial ecology research program in Baja California, where we brought in biogeochemists, people doing molecular biology, people interested in more geologic aspects, and organic geochemistry, to really get a holistic view of what a cyanobacterial microbial mat is all about. And so we've had a series of papers, and I wrote some review papers over the years of our understanding of that, and I think it came to be recognized at a point as probably one of the best characterized microbial ecosystems that were particularly relevant to our early biosphere. And I should add now, of course, that there's been a number of other environments now where people have done comparable amounts of work, but this idea of really getting a bunch of interdisciplinary efforts going at a focused place like that I certainly was involved in that in the early going, and I'm pretty happy about what's ensued from that. And then, of course, I should mention my role in the astrobiology roadmap. I chaired the major revision in 2003, as well as the update in 2008. And I think in terms of just trying to help to determine focus for the program, a few things that are key, I sort of alluded to them as I answered an earlier question. I think has been helpful to keep the ship sort of heading in a direction which the community really determined was the most important way to go. So these were not my ideas. My job was just to make sure that the community ideas were focused in a document in a way that would be useful for NASA headquarters to move out on. So those are some of the high points. There's more details, but I think in the interest of moving to the next question, I will do that. The most surprising or impacting discovery in the field of astrobiology, well, just that was your second question. I mean, everything I mentioned about Mars, that there's so much more widespread evidence for water activity on the planet early in its going. I mean, I'd be remiss if I didn't mention for exoplanets the major discoveries by Kepler. I mean, that's huge. I don't think even the Kepler people expected that they would have such a high discovery rate. That's a huge impact for astrobiology because it's all about habitable environments. So the Mars thing and the exoplanet discoveries by Kepler all address this very important habitable environment aspect of astrobiology. In terms of in terms of other aspects, I mean, I think every major aspect of astrobiology, there's been something that's really important that's happened in the last 10 years. The increase in the program that happened in the 90s has just led to a lot of this stuff. Ideas about the roles of RNA, interesting aspects about molecular evolution and gene transfer. A lot of that research was supported through the astrobiology program, and you can just add that to the list. I'm in real serious danger here of leaving out something, but I just say that there's more examples that could be cited. Aditya, I understand that every time we send spacecraft to Mars, there are always some bacteria spores that hitch a ride despite the cleanup process. Could you please describe if these are expected to survive space transit, EDL, and the environment of Mars, even if it is just for a short time? Okay, there's a serious effort to decrease the bioloading on these spacecraft. But because the ones that we've sent recently are not life detection, these loading reduction has not had to be as severe as it was for the Viking mission. So absolutely right. The bacterial loads are very low, but some of them do escape the cleanup process. Just one example I could give is that the circuitry, the solid-state circuitry that is used, you just can't heat that like we were heating the rather primitive stuff that was passed for electronics back in Viking. And so we're inevitably going to have some organisms in there. So they definitely survive space transit, they survive EDL, and they're probably lurking out in the rover for a period of time. But there's an important consideration here, and that is that the surface environment is hostile, okay? And so if an organism were to get away from that spacecraft, it would probably not survive for very long. And this is the key thing. This is what's called the loading rate for Mars, the ability of Mars to accept microbes coming from the Earth, but yet destroy them at a rate that's greater than the rate at which they can propagate. And so the way we've done these spacecraft, they definitely fall within the safe envelope there. Sure, there's bacteria on there, maybe a few, I'm not going to guess at a number. There's easy to get a lot of them. But, they're in that spacecraft. If they're on the outside surface, they're getting toasted. They're probably gone already, certainly on the two Mars rovers, Spirit and Opportunity. There are probably some lurking inside, but if they stick their little heads out, they're toast. So that's sort of the situation. None of this is proven, of course. You don't assemble a rover ready for launch and then tear it down again to do bioassays. But, there's ways at JPL and at Kennedy where they can do assays and get what they call a statistically representative sampling of what's in those rovers. But anyway, that's the overall picture. EDL means entry, descent, landing. Kara Tucker, what do you believe NASA's role is in education and public outreach? Gee, that's a topical question. Well, I think the principal role of NASA in education and outreach is obviously to give the public a return for their investment and to do things that really sort of advance the the interests of the country, the long-term well-being of the country, and of course that would be maybe principally in the area of technology development, but also in terms of an inspiration. I think science and technology are central to the economy of any country. Any country that is sort of ignores that is at their peril. And NASA has not only been excellent at having technology development happen directly as a result of its actions, but it's, I think, excellent at inspiring young people to consider, if not NASA as a career, some kind of a science or technology career. And the thing that follows from that is that I think it's appropriate for NASA to have the role of disseminating the information that we get from our research and from our missions to this first step of dissemination, to where an education type organization, such as perhaps Department of Education or the public school system or whatever, state school systems, could then take it from there. So I think really there needs to be a capability that's very close to the missions themselves that disseminates that information, or very close to the research programs such as the Astrobiology Institute to do that. I think that is a role that's totally within NASA, and I think that there's been a lot of consensus, not just within NASA but elsewhere for that. So I think for me that's the most defensible role. When you get out into the business of being in classrooms and so forth, well, many of us enjoy that, having direct contact with students. I guess there could be more of a discussion as how that type of activity would be partitioned between NASA and other agencies of government, both state and federal. So that's a first order answer. I think the first step towards education outreach, information moving out to the audience is a NASA role that's associated with the sources of the information, be they missions or some of the basic research programs. Okay, here's Kara's question. One last question for me. Any advice on how to survive grad school in planetary sciences? How to survive grad school in planetary sciences? Okay, well, the first thing that comes to mind is this comment I made earlier about maintaining sort of a core discipline, be it in chemistry or in physics or biology or something like that. And as you know, planetary sciences very much involves physics and mathematics. And of course, various aspects of Earth sciences. For example, the questions that we had earlier about remote sensing of Mars, or the comments I made about, the CRISM instrument or the Mars Express instrument, Omega, or just for that matter, the remote sensing observations of HiRISE and the camera on Mars Express. These are all aspects of geological sciences, environmental sciences that are used on the Earth. I mean, we do a lot of remote sensing of the Earth, and so geologic things are also very important for planetary sciences. I think the way you survive grad school is just make sure that you've got your courses and your learning curve definitely on track with respect to one of these core disciplines as you're going through grad school. Surviving, well, I mean, that's getting good grades. It's getting a good research topics that you can defend in your qualifiers. It's just like surviving grad school and any of the other topics that are more traditional. Planetary. I guess the other thing I should say about planetary sciences is that there's a number of departments around the country, and I guess it's really true just in biology or physics or anything else. These departments in planetary sciences, they have different emphases and specializations. And and that sort of gets back to what's your interest. I mean, what is it that turns you on about doing science or technology? That sort of then starts with, do you do physics, chemistry, math, or biology or whatever? But then it also relates to then what aspect of planetary science do you find compelling? And some schools are just really very deep well about that. They have a very deep well of opportunities for that. And so I guess what I'm going to finish with is the comment that you'll survive the best in grad school in planetary scientists if you're true to yourself and your interests. If you're really interested in something, you're going to do well. And that means making sure that you're in the right discipline, like chemistry, physics, biology, or whatever, and that you're also in this planetary science school that really resonates most completely with what aspect of planetary science you find most compelling for yourself. To Shauna Kendall, to expand on your background, it has become quite diverse. Did you just keep an eye out for opportunities as they came up? Okay, well, thank you for that. Actually, there's two kinds of careers. This is sort of a joke, but maybe not. And you can either have the animal or plant type career. The animal career, you're a specialist, right? You're really good at something, and to keep your resources and your lifestyle, you move around, okay? Or the plant is something that sort of sits in one place, but it has to be quite versatile in its chemistry and some of the things it has to cope with by being stuck in one place if environments can change. And so I've been here since 1976, and so you might say I'm more of the plant. And that is, over the years, I've seen opportunities come along, and I've gotten excited about them. Again, you got to be excited about it. And I've just sort of chosen to change my research emphasis over the years in response to changes in opportunities, but also things popping up that I said, I could get interested in that. And so I sort of took on the plant type mode of life. That might be a luxury these days. I don't know. I mean, I think a lot of young people who are very skilled realize that they maybe have to change jobs every few years to keep their core skill set, but tailor their strategy to changing conditions, changing environments, as it were. So I think there's sort of a biological analog here that's useful. I just happen to be fascinated by the way systems integrate with each other. So I was interested in this carbon cycle thing long before, the interdisciplinary astrobiology thing became a fashion. Well, more than that, it became a very compelling program. And so I guess just my interest in how systems work in concert set the table for me to be interested in how systems would work more in the context of astrobiology. So I guess for me, the fact that I've been involved in a number of things and over these years was sort of due to my receptiveness to being interested in how systems work together, and then also just being lucky, to working for NASA. I have to say it's just been rich with opportunities, and to have my interests and the opportunities here match nicely. A question from Hessian: if money and resources were not an issue, what would you like to see the next mission to Mars to include or accomplish? Well, I would like to see the ideal would be to have a curiosity type mission with maybe a little bit of a rock grinding capability there. That really would have the ability to get samples cached and to bring them back. I'm sure many of you heard all the arguments why sample return is such a compelling thing. And I guess, some people say, well, we've already got Mars meteorites. Why do we have to go get samples ourselves? Well, Mother Nature chose which samples to give us in terms of the Mars meteorites, but we get to choose which samples to bring back, if we actually go there with the mission. So I would sure like to see a curiosity capability put together with a sample return. That would be just wonderful. And of course, as you know, and I think a beautiful example of this was the Allen Hills meteorite debate and analyses that were done, is that you get a sample back and suddenly it tells you that there's things you could look at that you didn't even think of. And there's laboratories that you should get involved that never have been involved before with analyzing samples from other planets. And so just having those samples back, and in this case from a compelling place, to me, suddenly really puts us in the game of life detection on Mars. And with what we've now learned about where to go, and I think as this mission unfolds at Gale Crater, people will just get more and more amazed by just what a wonderful site it was for landing. To me, would be just tremendous. To have an intelligent site selection of samples, you put it together and get them back. I'd love to see them coming back in the 2020s. And just get on with that whole new level of investigation and, I think, understanding about Mars. And I should add that a sample coming back would include an atmospheric sample as well, and a sample of the regolith. Every time we've had samples come back before, it's just told us things we never even knew to ask questions about. And I just think that's going to be transformational for Mars science. Let's see, so that question was Mars. Okay, so we'll leave it at that. Okay, regarding manned missions, what do you think about the private sector's plans to send people to Mars? SpaceX.com. And should they work with NASA to planetary protection reasons? Well, I think it's wonderful. I mean, I think SpaceX in particular has shown that they can very economically get up to the space station and do things that, frankly, people didn't suspect were going to be possible so soon. And there's every reason to suspect that their approach could be one of a very effective approaches for doing Mars. My personal philosophy about sending humans to Mars is that the first mission would be an orbital mission with the humans, and that they could do a lot of interesting things with robotics, not just on Mars, but with maybe Phobos or so forth in orbiting the Mars. Be a lot less risk. It'd be a lot easier to envision them actually being able to come back. And it would retire a lot of risks associated with an ultimate mission to the surface. Now, some people are much more enthusiastic about going to the surface on that first mission. And, I sort of depart from that because I'm used to, doing missions in a series. And, of course, the classic example of that was the Apollo missions, where we actually really got there, but we did it in a series of deliberate steps. So, I think what the private industry is doing is wonderful. I think there's a difference of opinion about whether that first mission would be to the surface with humans or in orbit, because we have the whole planetary protection issue also with Mars. But I think it's a great thing, and it's our future. I mean, the private sector is going to be the future for all of this. It's just a question of when and how. What are the advantages, disadvantages of looking for biosignatures in the visible versus infrared while characterizing exoplanets? Well, let's see. Infrared, Infrared is better for, that's been like, 12 years since I really got involved in this. There's a lot of water lines in the visible that help with detecting water. The infrared gives you a bunch of molecular information that, it's really good because of the ozone, the O3. It's very strongly detectable in the infrared, so you probably have better sensitivity for oxygen. But you can see oxygen in the visible, but it's more challenging. So that's right. It's coming back to me now. Infrared is better for O2, and that is really maybe the ultimate biosignature that you could hope to detect remotely. There again, as I said earlier, this question was the source of the comment that it would be lovely to do both. They're very complementary with an E to each other. As I just said, the visible is really good for water. The infrared's better for O2. CO2 also better in the infrared. In terms of signal to noise, the visible is making rapid progress because of just the incredible improvements in the technology, and it has less troubles with the dust problem, the zodiacal dust problem, and so there, it's sort of TBD. Anyway, that's some comments. On a lighter note, what was it like to how did somebody find throughout the opening pitch at the San Francisco Giants ball game? That was quite an experience. You're a rock star right up to the moment you throw the pitch, so that was sort of cool. People interviewing you and all that kind of thing. I got some really great advice before doing it, and that is don't stand on the mound and on the rubber and try to throw it like a professional pitcher. Because one of the things they said that the crowd will do if you throw that ball and it hits the dirt before it gets to the catcher is that they will boo you. The entire stadium just loves the idea of booing people who throw the ball in the dirt. And he said, this advice I got said that the first George H.W. Bush threw it in the dirt in San Diego when he was down there, and the whole crowd booed him. So even the President of the United States will be booed. And he said, you can laugh it off, he said, but it'll stick in your conscience that the whole audience booed you. So he said, stand in front of the mound, right in front of it, and then throw that pitch. A nice firm arc, don't try to throw a fastball, and if you can get it to him in the air, you will have succeeded. So I did that, and it was really quite an amazing experience that way. There's a little story about a robot was supposed to come out and deliver the ball to me, and I had to wrestle it out of the claws of this robot, which sort of was going against the message about humans cooperating with robots. But that worked out, and I still have that baseball. It's got some dig marks on the two sides where the robot wouldn't let go. But other than that, it was pretty cool, and they give you a free ticket. And once you've thrown the pitch, they say, "Okay, get the heck off the field and get back up in the stands," and it's over. But it's neat, and I have to say, for me, the most enjoyable part of this has been the fact that people keep asking me this question years later. It's something I did that probably has more recognition than any paper I ever wrote. So thanks for the question. The tradition continues. Okay, and with that, I guess I'll wrap up. Yeah, and I think that's it pretty much for the questions. So it's really been enjoyable, a great set of questions. I appreciate it, and as always, when you get these types of questions, it compels you to think a little more deeply about not only the what, but the why, and that certainly was the case today. So thanks again.